All right, so on this activator, the idea is we start with the words. From the words on this activity that we used over like last week, we can go through three letters, we can take the word, we can do the three-letter sequence of tRNA and then kind of work backwards. So, like for example, the word start was UAC. The complement of UAC is AUG for mRNA, tRNA and mRNA. The complement of mRNA to DNA, now remember DNA has T's, not U's, so then it's going to be TAC. Okay, and I'm just going to kind of put this up there. And what I would do if I were you, I'd just kind of take a look at the DNA sequence. If you get these, and I didn't do it all because I kind of, I'll be honest, I got a little lazy. But if you get those right, I'm assuming you did the last couple right. So, but TAC, TCA, TAA, CAG, AGA, CCT, CCC. Now, if one of those is wrong, please let me know. I hopefully didn't screw anything up, but that's the idea. But then the complements kind of build upwards, right? So in a normal sequence, what happens is it goes DNA to mRNA. It's called transcription. From mRNA to tRNA, that's called translation. And then in our cells, that puts, instead of bringing words together to make a sentence, it brings amino acids, which makes proteins. Okay, so if we kind of take a kind of big picture approach to things, that is what I have written up on the board. That when we talk about DNA, DNA is genetic information that codes for proteins, but proteins express traits. So why is your hair color the color it is? Why is that trait for your hair color that color? Because you have a protein that expresses that trait. And why do you have that protein? Because you had genetic information stored in your DNA. That's like the big picture. DNA codes for proteins, proteins express traits, but there's a lot of little steps in between. So DNA is your genetic information, the master copy, that through transcription is made the disposable copy of mRNA. The complement of mRNA brings in the tRNA. On that tRNA, it brings in a very particular amino acid. Depending upon what amino acid you put in order, or I'm sorry, depending on the order you put your amino acids together depends upon what protein you make. And depending on what protein you make, depends upon what trait you have. So a lot of our examples we've been putting together, we've been using tRNA for words to make sentences, or we've been putting letters together to make words. But in reality, it's coding for amino acids, and the amino acids make proteins, which then make traits. But if something gets screwed up here, your amino acids may change, which changes your protein, which changes the trait. Just like in our examples, if you screw something up, your sentence might be different. You put a different word in there. Okay. Now, I do want to look at the questions underneath that chart on sending codes with DNA. Yesterday, when I was going through and I was grading those, I marked up a fair amount of them because there was a fair amount of mistakes on these bottom questions. So I went through and I tried to, and I would make note of what the mistakes that I marked, but the idea is that these are the correct answers. That when we have DNA to mRNA, that process is called transcription. Most of you, that wasn't the issue. The location where DNA is copied as mRNA is in the nucleus. And you can say, well, how do I remember that? Well, remember, DNA never leaves the nucleus, so if you're going to make a copy, it would have to happen in the nucleus. When you go from mRNA to tRNA, that's called translation, and translation occurs in the ribosomes, which is in the cytoplasm, but it's on the ribosomes is where that process occurs. A lot of you said, some of you said nucleus, some of you said cytoplasm, but it occurs on the ribosomes. And when we talk about 3 nucleotides in a row specifically, and it probably should have been more clear, every 3 mRNA nucleotides, that's referred to as a codon. So 3 nucleotides of mRNA in a row, A's, A's, U's, G's, or C's, because mRNA, no such thing as a T, that's called a codon. That's called a codon. On the top, this is what I kind of wrote on the board. It's that flow of genetic information. Now, it doesn't have DNA to mRNA, but mRNA codes for tRNA. Almost most people got this wrong, or didn't have what I thought was the appropriate flow of genetic information. Okay, so mRNA codes for tRNA, and tRNA codes for amino acids. That tRNA brings in a specific amino acid. Okay, so hopefully you're making these corrections. Most of you did okay on this bottom thing. This really weird looking thing is a tRNA molecule. This is a tRNA molecule. This thing up top is an amino acids. There's 20 different amino acids found in your body. The sequence of those amino acids put together make different proteins. Okay? This amino acid is specific to this 3-letter combination of A's, A's, U's, G's, and C's found on the bottom. These 3 letters are the complements of the codon. And what's the complement of a codon? It's called an anticodon. If you change the 3-letter sequence of the anticodon, chances are good that it's going to be coding for a different amino acid. Okay, so kind of keep those things in mind as you're kind of working through that. Anything I can answer about that before we move on? Okay, so the next thing I want you to do is over on, I kind of have it set up in a clockwise rotation. There's a packet that I want you to grab, and then I want you to grab a colored pencil from each cup all the way around the room. There's 8 of them. Okay, there's some light blues and dark blues. There's light green and dark green. It doesn't matter if you just need a blue and you need a green. So you're going to grab this worksheet on that table, and then there's a total of 8 cups. Grab a colored pencil from each cup, and then bring that on back to your desk. Go. 1 staple packet. Our class is a little bit smaller, so you can be somewhat selective on picking the colored pencils that actually have a point on them. They don't have to be super sharp, they just got to be able to kind of do a little bit of coloring. One packet, 8 colored pencils. All right, so we have the packet called Protein Synthesis and Amino Acid Worksheet out. If you're working on this at home with us, maybe you're marking up the PDF so you can use your markup different colors on there, or if you choose to print this out, and hopefully you got some crayons or colored pencils lying around at home. Okay, so the idea of this is we're going to have a nice, at the end, a nice kind of color-coded version of what DNA structures look like. Now, some of these we've seen all of this before. The fact that we've seen this stuff already is a pretty good indicator that this is important things. So when we talk about a nucleotide, remember a nucleotide is a monomer, and I'm just randomly using the color brown. This is not specific to the color brown, it's just what I grabbed. But nucleotides are the monomers of DNA and RNA. And I've asked you to label this picture multiple times. There's 3 parts of a nucleotide. This is our nucleotide. There is a phosphate, a sugar, and a base. So of this, the phosphate is the circled thing. Okay, so I'm just going to do a kind of quick color with my brown. Okay, so my brown is the phosphate, and my phosphate is the circle. Now, if we kind of go down, we're going to use the same color coding down below. All of these all of these circles are also going to be shaded in brown, just doing some quick coloring, because that's my sugar phosphate backbone. The sides of my ladder, those are my browns. Okay, that's my phosphate group. There is a 5-carbon sugar. Now, what is the sugar in DNA called? Deoxyribose is the sugar in DNA. And what is the sugar called in RNA? Just ribose, yep. So if this molecule, this monomer, this nucleotide is DNA, then this sugar would be deoxyribose. The pentagon represents the 5-carbon sugar. So my pentagon is orange. All of these pentagons, in fact, on my molecule are orange as well, because those are the sugars in our sugar phosphate backbone, right? Sugars, phosphate, sugar, phosphate, sugar, phosphate. So those are all of our sugars. So the browns are the phosphates, the circles. The pentagons are the 5-carbon sugars, are in orange. And notice how I'm doing both them up top and down below. And the last part of our nucleotide consists of, those are browns, that's our oranges, and our purples. Okay, and our purple is this base. Okay, and in general, for a nucleotide, I'm going to use this purple up top. That is our base. If it's DNA, it's an A, a T, a C, or a G. If it was RNA, it'd be a U, an A, a C, or a G. Okay? Now, based upon the color coding over here, we have different colors that we're going to use for different bases. Okay, so we have 4 different bases. Now this must be DNA, and I know it's DNA because it says thymine. Now we went through and we gave you this is a C and this is a T. Now based upon this being a C, what would have to be across from it? A G, right? So this is a G. And if this is a T, then across from it must be a A. Now, using kind of our shapes, okay, we can go through and we can say, oh, are there any other bases that have that shape? And if so, we should put a letter C in there. Well, this is the exact same shape as this one. This is the same as this letter C, but it's on the left-hand side now. And then this one is also going to be the letter C. You guys all see where I got that? That shape is the same as that shape, which is the same as that shape, which is the same as that shape. Now, according to that, what was that guy's name? And he had that one rule about complementary base pairing. It was like a CH something. What was his name again? Charga! Charga, very good, very good. And he said for every C, there's a G. So there's got to be G's across from it. And kind of using our same template of, hey, this A has kind of a point sticking off of it. This is going to be an A, and according to Chargaff, every A there's going to be a T. And now, using kind of our color coding, just to kind of give us a nice visual representation for every A, we're going to color those in red. For every A, we're going to color that in red. My T's are all colored in green. Notice I'm not doing an amazing job coloring, but Mr. Davies, you color very nicely. Oh, thank you. You're doing a wonderful job, I'm sure, as well. And for every C, we're going to color those in blue. The idea is getting some color coding, nice visual representation. That's my C's, and all my G's are going to be colored in yellow. Now, notice how I drew these arrows in, and I drew them in opposite order. That's trying to kind of reiterate the point of anti-parallel. So notice how up here there's a phosphate, sugar, phosphate, sugar, phosphate, and then phosphate, sugar, nothing. And then down here we start with a phosphate, sugar, phosphate, sugar, phosphate, sugar. Now here there is no phosphate. Okay, that's that anti-parallel. Now, for this to be a better model, what really should I have done with probably these letters over here? I should have, I'm sorry, what's that? Yeah, so I would have probably written them upside down, right? But that's the idea, because they do flow anti-parallel, just like we did with our DNA structure. Okay, so that's the idea. Okay, we have lots of sugar phosphates. These are backbones. Okay, this is sugar phosphate backbone. These are the rungs of our ladder that the bases are held together. And what kind of bond holds those bases together? Hydrogen bond. Is that a strong bond or a weak bond? Relatively strong or relatively weak?
Relatively weak.
Yeah, it's relatively weak, and that's what holds these things together. So notice how there's a little gap in between our base pairs. That's what we're going to use that black colored pencil for really quick, is we're just going to kind of go through and we're going to fill that void, that space, with our black colored pencil, and the intent is that's a hydrogen bond that's holding those two things together. Relatively weak bond. Can be broken? Absolutely, it can be. It's broken when DNA is replicated, or we're doing transcription to make mRNA. So that's what page number 1 on this activity should look like. Nice color-coded thing. A brown, an orange, and a base makes up a nucleotide. Now we have 4 different types of bases. Could be a C, could be a G, could be an A, or could be a T. But we have a bunch of nucleotides. DNA is a single strand or double strand? Double. Double strand. Okay, and it has the letter T's in it for thymine. Whereas mRNA is single strand and will have U's, but we're going to get to that in a moment. Okay, any questions on that page 1? All right, then let's turn the page, and we're going to take a look at this. Now, this is a process of transcription and translation. There's a DNA strand, and it's asking you to make the complement or write the complement of the DNA. So we're going from DNA to DNA. Okay, that's Chargaff's rule. Then we're going from DNA to mRNA. That's called transcription. And then we're going from mRNA to tRNA. That's called translation. Now, what happens is these 3 letters of tRNA bring in a very specific amino acid. Okay, so that's why this is kind of written like this. 3-letter sequence of mRNA is called a codon. The complement of a codon is referred to as an anticodon. So mRNA is the codon, tRNA is the anticodon. And that tRNA, the sequence of the bases brings in the very specific amino acid. So we change the sequence, we change the amino acid. Now, we got to be careful when we code for amino acids that when we look at the charts that we are coding it off of the right thing. Okay, so we'll get to that in a moment. So DNA to DNA, that's really all we're doing. This is following Chargaff's rule right here. Okay, for every A there's a T, for every T there's an A, for every C there's a G, for every G there's a C. So if the original DNA strand is ATG, the complement is TAG. If the original strand is GTA, then what is the complement of that DNA strand? GTA, the complement of DNA is going to be CAT. Good. Because it's DNA, there is the letter T. If the next sequence is GCT, what's the complement of GCT? C, what? GA, yep. Now, I made a video for the A-Day students yesterday, and I screwed that up. Okay, make sure that you do the complement. It's easy for there to be a mutation. Okay, so just kind of keep that in mind. There are, what, 6 more base pairs? Do the next 6 DNAs on your own. What's the complement of AAC? What's the complement of T, whatever the sequence is? I don't have it in front of me. Whatever the next thing. So you do these 3 DNA, and then you do the next 3 DNA. Chargaff's rule, complementary base pairing. On page 2. So AAC, the complement of that is TTG. CTT, the complement is GAA. Any questions on that? What's that? Okay. If I screw something up, please ask, because I do it from time to time, but just keep in mind that there are T's in DNA. Now, the process of going from that DNA to mRNA, that next piece is called transcription. Now we're going to RNA, and in RNA, there was no such thing as a T. So if you would have put a T in, we put a U in instead. So that's called transcription. So in the top 3 that have already been filled in for you is TAC was their DNA. The complement of TAC is AU, right, because there's no such thing as a T, AUG. What is the complement of, now we're going to RNA, remember, of CAT. The complement of CAT is? Not GA, GU, right? For every A, there's a U, and then GU, what's the third letter? A, yep. So substitute that letter T. If you would have put a T, now you put a U instead. CGA, complement is? GCU, good. You do the next 6. All right, the complement of TTG is AAC, and the complement of GAA is CUU, because there's no such thing as T's in our RNA. That process is called transcription. That's called transcription. Now to go from DNA to mRNA is transcription, but from mRNA to tRNA is called translation. The 3 letters of mRNA are called codons. The complements of that on tRNA are called anticodons, just like it's coded in, it says in the column title. Okay, so the complement of AUG is UAC. For every A there's a U, for every U there's an A, for every C there's a G. I'm sorry, for every G there's a C. So what is the complement of GUA? C A U. Yep, C A U. What's the complement of GCU? CGA. Good. Do the last 6 on your own. We're going from DNA, I'm sorry, from RNA to RNA. So there's U's instead of T's. Keep that in mind. We have U's instead of T's. All right, AAC, the complement should be UUG, and the complement of CUU should be GAA. Okay, questions on transcription or translation, complementary base pairing? Okay, then let's take a look at the codon chart found on page 4.
Yes, page 4.
On the very, very back page, page 4, there is this thing that is referring to as a codon chart. Now, there's different things we can code off of. We can code off of DNA, we can code off of mRNA, or we can code off of tRNA. So each time you have a chart, this chart's a little bit different than the charts we've seen thus far, but make sure you know what it's coding off of. Is it based on mRNA, tRNA, or DNA. Now this one I feel is somewhat clear by saying codon, but it's not as explicit as I'd like. But codons are found in mRNA. So this chart codes off of an mRNA sequence. This is not tRNA, this is not off of DNA, this is off of mRNA because it says codon. Now this circle chart is coding for amino acids instead of letters or words in a sentence. The idea is you start with it's a 3-letter sequence. You start with your first letter in the middle, and you kind of just keep working outwards. So like your first letter is in the first ring, and then the second letter, then your third letter, and then your amino acids are all the way around on the outside. Now there's a chart on the bottom that we have 3-letter abbreviation, a single-letter abbreviation, and then the full names. We're just going to use the 3-letter abbreviations for today. But realize they stand for a specific name. So the idea is we start, like if it was UCA, it'd go UCA, and then it'd be that amino acid. So our first one, we're coding off of mRNA, remember. So we are coding off of the mRNA column, so it's going to be this column right here. The first 3 that is already done for you is AUG, right? The very top one that was already filled in for us was AUG. So if we look up AUG, it says that we should get MET or MET or methylene or the start codon. So the idea is here's my first letter, and then we work outwards. So AU, now we're in this ring, and then the third and final letter is that G, and then our amino acid is MET or methylene, or there's a little arrow, that little triangle is coded for start. So we have 3 stops and 1 start codon. So that's how this chart works. And this is a chart that you're going to use in your homework for tomorrow's virtual day. So being able to use this is important. It's pretty, generally, people don't tend to struggle too much with this one, but make sure that if you don't understand that you ask.
Start in the middle.
Middle, and then just keep working outwards. So what letters are they, right? And then you get to the outer ring. So the next 3-letter sequence is GUA. So we start in the middle at the G, then we work outwards to U. We got to find, oh, there's the U, and then it was, what was the last letter? A, thank you. GUA, and then that codes for val, VAL. Now, according to this, there were 4 different combinations that would code for val in that triangle, or in that kind of vector kind of direction. And val stands for valine, or we could use the single letter abbreviation of V. We're just going to use the 3-letter abbreviation for us. We're going to use the 3-letter. So val is what we have. Remember, mRNA, the next 3 mRNA sequence is GCU, GCU. So GCU, oh, that's going to be ALA, or I don't know how you pronounce that, ALA, ALA. Okay, so that would be our next amino acid. There's 2 more sequences for you to code for. You do the last 2 amino acids on your own. Code for the last 2 amino acids on your own.
You're coding for AAC and CUU.
While you're working on that, I'm going to come around and I'll collect your colored pencils. Okay, you can just put your colored pencils right in the tub. Coding for those last 2 amino acids. All right, so what's the next amino acid? I don't know because I'm not looking at it. What's the next amino acid? ASM? That sound right?
ASN.
Okay. ASN. And the last one? Leu, L-E-U, right? Okay. Any questions on how this chart works? We're coding off of mRNA. This chart codes off of mRNA. Okay. There's one more sheet that we are going to get started talking about, and it's either found on this cart right here or right next to that box of colored pencils. Maybe some of you grabbed it already. It's called How DNA Determines Traits Lab. Grab one of those. So if we look at the string of sequencing on the board for genetic information, it says DNA codes for proteins, codes for traits. Or if we kind of break down even further, DNA codes for mRNA, that's transcription. RNA codes for tRNA, that's called translation. And then that tRNA sequence brings in a particular amino acid. Depending upon what amino acid comes in, will help determine what protein is being made, and based upon that proteins express traits. So what this activity is looking at is how a sequence of amino acids are going to code for a particular trait. Okay? So in the activity called How DNA Determines Traits Lab, a distant alien planet similar to Earth has been discovered. The most common species on the planet is called a unigriffin, which is a hybrid of birds, lions, and unicorns. Scientists have recently obtained DNA samples and have mapped out 8 genes so far. Your job as science students is to analyze the DNA sequence of the unigriffin samples to determine which features each sample codes for, determine which traits each type of unigriffin has by decoding the DNA. There are a total of 8 genes that could be with 2 possible versions. So notice how we have 8 different traits. We have tail, lion's mane, unicorn horn, eye color, beak, front legs, wings, and ear shape. And then there's a description of them. So there's 2 different choices for tail. There's 2 different choices for each of the different traits. Okay? Now, if we look at page 2 and 3, I want to draw your attention to the fact that there is 3 different samples. There's 3 different organisms. There's the unigriffin, there is the unique unigriffin, and there is the unruly unigriffin. Okay, so there's 3 different unigriffins that they've got DNA samples from. And what's going to happen is we have 8 different sections of DNA per organism, right? So here is a chunk of DNA right here. Here's a chunk of DNA, here's a chunk of DNA, DNA, and there's 3 more on the other page. So there's 8 different traits that each unigriffin is going to have, and then they're going to tell us a particular thing. So let's work through this. Our DNA sample is CAT, AGG, GAC. What is the mRNA sequence that's the complement? What's the transcripted mRNA sequence for CAT? GUA. Okay? GUA. Now, there is no start and stop codon on these, just realize, but there just isn't. The complement of AGG is, what's the complement of AGG? We're right there. What's the complement of that? UCC, and then the complement of GAG is CUC, right? That's our mRNA sequence. Now, this chart right above it is the same chart that we were just looking at. It's mRNA. This is coded off of mRNA, and it does say it up in the description that this is an mRNA sequence. I don't even ask you about tRNA in this particular activity. So the idea is if we start with the letter G, so I'm looking at these 3, right? GUA. So I start with G, then I go to U, and then I go to A. My 3-letter combination of my amino acid is VAL. The next 3 letters are UCC, UCC, it's serine, SER, and CUC, CUC is leucine, which is LEU. Okay, so we have a 3 amino acid sequence. Now we can go back to page 1, and based upon the sequence of Val, Ser, Lu, we can go through and say, oh look, Val, Ser, Lu, the ear shape of the whatever Ula unigriffin has rounded ears like a lion. So that amino acid sequence codes for rounded ears like a lion. Okay. On the back, on page 4, notice how we have 3 different templates for you. And Ula unigriffin, the idea is we just determined that based upon that 3 amino acid sequence that it's going to have rounded ears like a lion. So when you draw, yes, you will draw your unigriffin. Your unigriffin for Ula unigriffin should have rounded ears like a lion. Whatever that means, I'll be honest, I'd have to Google it to look up what a lion's ears look like.
Oh, it's
rounded.
You know, artistic license here.
Artistic license, yes. Okay, but they should be rounded, not pointed. That is the idea, right? They have rounded ears, not pointed, because that was the other version of that trait. Okay? And then we would go through and we would look at whatever the next trait is. Okay? So are they going to have, whatever the sequence of this is, goes to this, that goes to this, and then whatever my amino acid sequence is on the front, we'll talk about, I'm assuming they go in order, we'll talk about are the wings of this Ula unigriffin large or are they small? The next one is probably going to deal with the front legs. So are they scaled with claws like a bird, or are they going to furry with claws like a lion? Or is the beak going to be short and pointed, or is it going to be long and curved? Okay, there are 8 traits for each unigriffin that you will decode what they look like. Now notice for eye color, it says green eyes and brown eyes. Ideally, you have green and brown color, pencil, crayon, something that you can use for the eye color. If that's not an option, then you should probably just draw an arrow to the eyeball that says green, so we know that it's a green eye versus a brown eye. So just be aware of that. But that's the idea of your virtual day assignment. You're going from DNA to mRNA, that's called transcription. Then once you have transcribed, you're going to determine the amino acid sequence, and then from the amino acid sequence, you're going to determine the traits or what they look like. That's your virtual day assignment for tomorrow. Okay. With that, I think we are, yeah, we're good. So if you want to stick around and ask a couple questions, you're more than welcome to. If you want to pack up your stuff and wipe down your desk and be on your way, that's also something you can do at this point. Have a great day. If you have any questions about unigriffins, ask.
Don't Google it. Good? Davies?
Oh, yes, thank you. Yeah, we should.